welcome back to the Wilson Forum, everyone. You know, the convergence of engineering and medicine and healthcare in broader terms has really been explosive over the last perhaps 20 or more years. We heard some of that earlier in this series when Bob Langer spoke to us about messenger RNA and the work that he'd done at Moderna in developing a COVID vaccine, which was a benefit to society very broadly. This whole effort has produced this convergence of engineering and medical care has produced some really remarkably valuable understanding and products that have really served society well. Today, we extend that principle of using engineering to serve people with our guest, Pete Searson, who is a member of my extended family of academics at MIT. One of the things I really treasure about having been an academic is having people like Pete in that family. You know, it's fun to watch them grow up, and watching Pete has been a special pleasure because this guy has transitioned from being a material scientist to someone who is one of the pioneers of tissue engineering. And it's just, for me, Pete, this has been a marvelous journey to watch. And I'm just delighted to have you here today and to talk with us about the work that you're doing at Johns Hopkins. Pete is a member of both the Material Science and Engineering Department and the faculty of the Biomedical Engineering Department at Johns Hopkins. He's also the Reynolds Professor at Hopkins. So lots of credits, lots of good work. And Pete, I'm just delighted to see you. Welcome back to the Boston area, even if remotely. And I'll turn the floor over to you.
Thanks very much, Ron, and hello to everyone. I'm delighted to be here and see a bunch of very active and curious people. You guys are what I aspire to be as I'm approaching the end of my career now as well. So let me just share the screen here.
Yep, good.
You guys can see everything? Okay,
Yep,
so
perfect.
I'm going to talk about the blood-brain barrier. What is it? What does it do? And why you should be interested. So a lot of this is going to be sort of relatively descriptive because my main purpose is to just try to get you interested in the blood-brain barrier and understand why it can play an important role in your life. Okay, before we get into details, I'll tell you a little bit about me. I'm an immigrant. I come originally from England, and down at the bottom of England down here in a small village called Hythe. So Hythe was incorporated in 1293. The village first appeared on the parliamentary rolls in 1293. And it is a small town now, population 20,000. This is the main street that you can see here. I was born at Hythe Medical Center, illustrated here. Not quite Mass General, maybe a different end of the spectrum, but it got the job done nonetheless. When I was a youngster, used to go and drink beer at the Fleur de Lis, which has a continuous record of serving beer since around 1100. And if you look at the map here, you can see that Hythe here is on the edge of a very large green area. That green area is called the New Forest, and it was established in 1079 by William I. So one of the ironies, I think one of you mentioned some of the events over the last few weeks. So one of the ironies could be for me is that England could become the 53rd state, I guess, after Greenland and Canada. So could be a massive reversal of my heritage here, potentially. Okay, so I came to Boston in the 80s and joined Ron's lab and spent many years in Ron's lab, which was a really transformative time for me. It's where I learned and developed an excitement and broad interest in science in general, and learned from Ron sort of to be very open and broad in one's thinking. And that's certainly been a major shape on my subsequent career. In Boston, when I first moved there, I lived in Peterborough Street in the Fens and learned a lot about baseball because we were only maybe a 5-minute walk from the baseball stadium. There was the one thing I remember there was a pitcher at the time called Oil Can Boyd, who was a pitcher for the Red Sox at that time. I also remember that hot dogs and beers were like $5 each, I think, at the time, which I'm sure is a lot different now. For the last few years in Boston, I moved out to the suburbs in Brattle Street in Arlington. And the cold weather, I think one of you mentioned, Tom maybe mentioned in North Carolina, it's also very cold here. I learned to dive while I was in Boston, and I did my certification dives at Marblehead between Christmas and New Year. So it was cold. That's all I remember about that time. Okay, so that's a little bit about me. Before we get into some of the details, there's 3 important definitions just to make sure we're all on the same page that I want to make. So first of all is the term cerebrovasculature. So the cerebrovasculature is or represents all the blood vessels in the brain. So you may have seen some of these art installations that have been done where human bodies have been displayed with certain parts of the tissue either dissolved or whatever. So this is an example of the all the blood vessels in the brain if you remove all the brain cells and other tissues here. The next important term is the endothelium. If you're not familiar with it, the endothelium is the layer of endothelial cells. These are the cells that form the lumen of all blood vessels in the body. And then the blood-brain barrier, which is the main focus, this is an interface. So those of you who are physical scientists, you can think of this literally as an interface between blood in circulation. So the lumen is just the part of a blood vessel where blood flow occurs and the surrounding brain. And this dark region here, you can see the very dark region is the nucleus, and the slightly darker region that around the perimeter of the blood vessel, this is one of the endothelial cells that form the blood-brain barrier. Okay, so that's basic definitions. So next, there's 2 important facts we need to know about the blood-brain barrier. And the first is related to the brain itself. So it takes 20 watts to run a brain. So the brain is one of the most energy intensive organs in the body, despite the fact that there's no moving parts. So unlike the heart and lungs, for example, and the power for the brain, obviously, or the energy comes from oxygen and glucose, and the brain consumes, as it says here, 15 to 20% of all oxygen leaving the heart and 15 to 20% of all oxygen consumed daily. So in the brain, we have 100 million neurons as well as many other supporting cells. They are power hungry, and there's a lot of them. And obviously in the human body, we've evolved so we have on-demand energy supply. So there's essentially no storage for the brain. So we have onboard processing units. So the lungs basically take oxygen and put it into circulation, and the GI tract takes food and puts glucose into circulation. So the energy required to power the brain is on demand. It requires blood being circulated through the body and through the vasculature in the brain. So as a result of these evolutionary decisions, all neurons have to be very close to a capillary, a small vessel in the brain, because they need to get their oxygen and glucose. So typically they're within 10 microns of a capillary. And because we have a lot of neurons and they all need to be near a blood vessel to get their source of energy, there's around 600 kilometers of blood vessels in the brain. So it's a huge, huge network. I should also say, I didn't say before, if you have any questions, please jump in. I'm happy to field any questions as we go along. Okay, the BBB fact number 2 is that the brain needs protection. So the brain needs to operate in a very controlled and tightly regulated environment, very different from the other organs in the body. So all our other major organs, heart and lungs, can operate in much more messy, if you like, environments. The brain needs to operate in a very tightly controlled environment and needs to be protected, as it says here, from fluctuations in salts and hormones, for example, that occur after eating or exercising. This can disrupt signaling in the brain. The brain needs to be protected from immune cell transport. So the brain is in a fixed volume defined by the skull, so we can't afford any inflammation. That generally will lead to increased intracranial pressure and edema, which is not good. And then the brain needs to be protected from toxins and pathogens that can also lead to inflammation and neuronal cell death. So these are 2 important facts you need to remember: that the brain is very power hungry, requires a lot of oxygen and glucose, and it needs to be protected from fluctuations in small molecules, immune cell transport, and toxins and pathogens. So that's sort of the essence of the blood-brain barrier, why it's called a barrier and why it's important to make sure that the brain operates in a tightly regulated environment. So if we look at the small vessels or capillaries in most of the body, most capillaries are designed on purpose to be leaky. So here we see a cartoon of a small capillary. The junctions between cells, so these little red lines here where cells meet, they're designed to be leaky. So there's paracellular transport. So transport molecules in circulation can leak out between cell junctions. And many small capillaries have, or the cells have, pores in them allowing transcellular transport. So molecules can cross cells through these small pores that are within cells. So in the brain, obviously, this wouldn't work because this would lead to entry of blood components. So all the things I just talked about that the brain needs to be protected from, at least from the small molecule point of view, this wouldn't work in the brain. So we're going to go backwards a little bit here with a brief history of the blood-brain barrier. So this takes us back to the late 1800s and early 1900s. So Paul Ehrlich, a German scientist, a Polish scientist, Max Lewandowski, and one of Paul Ehrlich's students, Edwin Goldman, in the early 1900s, performed a series of experiments. And basically the experiments boil down to this. So they take a small animal and inject a dye into circulation. And they found that that dye would stain all the organs in the body except for the brain and the spinal cord, so the central nervous system, broadly speaking. And eventually, Edwin Goldman, Ehrlich's student, did the so-called reverse experiment. He injected dye into the subarachnoid space, so into the brain effectively, and found that the dye stained the brain, but not other organs in the body. So this is the genesis of realizing that there's a barrier between circulation and the central nervous system. So this means that the blood vessels in the brain are not leaky like other blood vessels in the body. And parenthetically, it's a little note down the bottom here to remind me that Paul Ehrlich won the Nobel Prize for Medicine in 1908. He was way ahead of his time. He predicted autoimmunity. He coined the term chemotherapy, that obviously is in common use now. And he also came up with the concept of the magic bullet, which is still a holy grail for a lot of drug treatment in delivering a drug to selectively to a target. So very prescient scientist. Okay, so we know that the brain needs oxygen and glucose. It needs many other small molecules. So it needs amino acids because it needs to be able to build its own proteins, for example, and other small molecules. But the brain also needs to be protected, as we've said before. And we're going to talk now about the machinery of how the blood-brain barrier regulates transport into and out of the brain. How does it implement tight control over what gets in and out? So broadly speaking, there's three categories we're going to talk about here: tight junctions, efflux pumps, and transcellular transport systems. So this is going to be the most detailed I'm going to get, but I think it's useful to know. Okay, so here in the top left, we have our transmission electron microscope image of the cross-section of a rat brain capillary. And we've seen this image before, and indeed, transmission electron microscopy was hugely important in the history of understanding the blood-brain barrier. So back in the 1970s in particular, when TEM started to be widely available, many pioneering studies were done, like the images I'm showing, similar to the images I'm showing here, that provided huge insight into the blood-brain barrier. So what I want to focus on here, I've already highlighted that there's an endothelial cell that forms the lumen, that's the interface between the circulation and the brain here. And if you look at the region that's circulated here, the cell is lining the capillary and at one point is overlapping with itself. So you can just about make out this overlap region right here. And this overlap region is forming a so-called tight junction that's preventing leakage between where the cell membranes meet or where two different cells meet. So if we jump down to the bottom here, we don't really need to know the details too much, but where the cell membranes meet, there are proteins in the cell membrane that recognize each other and bind together, forming so-called complexes, forming so-called tight junction complexes or particles. That's illustrated in this cartoon here. So here's the overlap between two cells where their cell membranes overlap, and these red circles represent where these proteins bind together on opposing membranes to form these tight junction complexes or particles. So if we translate this to 2D now, this is the overlap of two cell membranes. You can see these lines of pinning points in this cartoon. And here we see a transmission electron microscope image of this cartoon, where you can see these white dots here are these pinning points or these complexes where these proteins on opposing faces are binding together. So it's sort of like, you can think of it as like riveting, if you like, the two cell membranes together to prevent leakage between the junctions. So if we jump back up to the top now, this is a high resolution TEM image. So this is a small part of a very small capillary. And the region at the surface here is the endothelial cell. And we can see the overlapping region right here. And in this particular experiment that was common at the time, a particular protein was injected into circulation into the rat in this case. And the protein was labeled with a contrast agent for electrons so you can visualize it in EM. And hopefully you can make out that there's a dark region in the gap in the interface between these two cells that runs all the way down to this point here. And then beyond that point, none of the protein is penetrating any further. So this is the brain side, this is the blood side, and this again is an illustration of how these selective proteins that I mentioned knit the two cell membranes together to prevent any paracellular leakage. So no small molecules can penetrate that region.
Excuse me, is there any way you can make the cursor a little larger? It's sometimes very difficult to follow on the diagram.
Okay, sorry about that. Let me see. Let's see if I can even make that bigger. It looks like the laser pointer is only one size here. I've gone. Sorry.
It's visible, but very hard, very hard. That's better. That's a little.
Is that better?
Yeah, it's red. It's more visible. That's
okay,
better.
I'll try and not move it around so quickly as well. That probably
Okay.
doesn't help.
Before you move on, I cannot resist but asking about horseradish peroxidase. Where did that name come from?
That's a good question. I don't know the answer to that. I'll look it up and see if I can find, and I'll email you afterwards.
Yeah, that's a deal. Thanks, Peter. I'm just curious.
That's I'm just making a note so I don't forget.
Yeah, okay.
Okay, so part of the machinery number one, so these tight junctions closely knit the cells together so they don't allow anything to leak between them. Okay, we're going to get a little bit more complex here. So here is our cartoon of what we've just been looking at in a transmission electron microscope image. And if we look at some region of the endothelial cell that forms the blood vessel here, so this is expanded here. Here we have the blood side. Here is the cell membrane that's in contact with blood. Here's the inside of the cell, the cytoplasm, and here's the membrane on the other side of the cell. So this is a single cell. So the other way that small molecules can enter the brain is some molecules can passively diffuse across the cell membrane. So you can think about this as a transport problem. And the transport problem is complicated by the fact that we have essentially water here in blood and water inside the cell, but the cell membrane is made up of lipids and has a hydrophobic core where the relative permittivity is very low. So for a molecule to diffuse across the cell membrane, it needs to have a balance of being soluble in water, but not so hydrophilic that it will be completely excluded from the core of the cell membrane. So there's rules known that describe what types of molecules can do this. There's only very few in reality. So some molecules are able to passively diffuse across the cell membrane, through the cell, and then across the cell membrane on the other side into the brain. And this is how oxygen is a gas, CO2, and a few other small molecules are able to enter the brain in this way. But we've evolved a system to prevent that happening for a lot of molecules that can diffuse across the cell membrane. And the mechanism is called efflux. So there are proteins that sit in the cell membrane, as illustrated here, that look like a clothespin, that are substrates of many, many different types of these molecules, and they will pump the molecules from inside the cell back out into circulation. So there's different types of these proteins. They tend to be multi-spectrum, and they pump a lot of these types of molecules out of circulate or back into circulation if they're able to diffuse across the cell membrane. So as a result, estimates have been made that only 98% of small molecular weight drugs are able to cross the blood-brain barrier by this method. So some examples of molecules that can do this are general anesthetics like propofol, morphine, and maybe some molecules that you guys are familiar with, things like caffeine and alcohol, are examples of molecules that are able to passively diffuse across the cell membranes and are not substrates of the efflux pumps. So together, the tight junctions and the efflux pumps are the reason why it's really difficult to treat diseases of the brain, because it's really difficult to get drugs into the brain. I'm not going to talk about that today, but that's another major research effort that's going on in our lab and other labs as well. Okay, so we have tight junctions that prevent things leaking between cells. Any small molecules that can passively diffuse across the cell membrane, most of those are returned to circulation by these efflux pumps. Okay, the third general category of the machinery is more related to then how do essential molecules get into the brain. If they can't get in between cell junctions and they can't passively diffuse, how do essential molecules get into the brain? So here we have a cartoon again. This is of an endothelial cell. Here's the membrane on the blood side. Here's the cell membrane on the brain side. Again, we have proteins that sit in the cell membrane. And this class of proteins, I'm just going to talk about one of them here, are called, or the process is called carrier-mediated transport. So these are proteins that recognize molecules and transport them across the cell membrane. So they're not limited by passive transport into the cell, and then transporters on the other face transport them into the brain. So this is how glucose, for example, the key fuel to run your brain, gets into the brain. So glucose is in circulation. It's recognized by these proteins. It binds to the protein transports it through inside the endothelial cell. It then binds to the same protein on the other side that transports it into the brain. So there's a variety of these types of machinery that are highly specialized and highly specific to make sure that only things that the brain needs gets into the brain. Okay, so I know that was very detailed, but that's the most detailed I think I'm going to get. But it's important, I think, to have some idea of what the machinery is that is regulating transport into the brain.
Pete, I have a question. I'm very interested in what you've just described. Is it conceivable that some of the, for example, some of the aging phenomena that we're familiar with is associated with environmental effects related to transport of species that are not ordinarily transportable? Is that possible?
Yes, it is. That's actually a very good question. It's a little bit beyond what I was going to go into, but yes. So the biological term is brain homeostasis. So the ability for the brain to work properly at all times requires all this machinery to work correctly.
Right.
So any perturbations or stresses on this machinery can result in all the things I talked about that we don't want to happen in the brain happening in the brain.
Yeah.
So
Yeah.
I'll come to that in a minute in a
Okay.
few slides, and hopefully that'll address your question. If it doesn't, then let me know.
Deal. Thank you.
Okay, so this is sort of actually setting the scene. So actually that was a good, your question was a really good setup for the next statement. So the next thing we need to talk about is the so-called functional interdependence between the blood-brain barrier and the brain. So in general, anything that promotes blood-brain barrier health promotes brain health, and anything that results in blood-brain barrier dysfunction can lead to brain dysfunction for the reasons that I've described. So blood-brain barrier dysfunction can lead to loss of normal brain function, either transiently or permanently. It can be local or regional or more pervasive. And again, we'll talk about some of the details in a little while. As we've talked about, if these parts of the machinery don't work properly, there's potential for entry of blood components into the brain, potential entry of pathogens into the brain or immune cells into the brain. This can lead to neuroinflammation, oxidative stress, neurotoxicity, and ultimately could lead to cognitive impairment. Okay, so we're going to look at one example here, or one type of example related to if the tight junctions don't work properly. So again, these are the tight junctions are the proteins that knit together, knit cells together to prevent leakage between the cells into the brain. So any stress that can lead to a decrease in those tight junction complexes can result in paracellular leakage. So that's illustrated in this cartoon. If there's fewer of these tight junction complexes, then it's possible for components in blood to leak into the brain. So, like everything, I'm going to preface my remarks by saying that nothing is ever black and white. So there's no hard and fast. It's not like everything works 100% well and any type of dysfunction can lead to bad outcomes. There's a complete gray scale. So whether one sees symptomatic responses to particular types of dysfunction is dependent on an individual's ability to tolerate it, depends on whether it's transient or persistent, and all kinds of other things. So I just want to make sure that's clear before we continue. So down here in the bottom left are shown MRI images of two individuals. So the first one is a 68-year-old male with early Alzheimer's disease. And in this particular imaging study, the patient is injected with a gadolinium contrast agent. And what we're looking at in MRI is gadolinium that's leaking out of the blood vessels. So that's this case shown in this cartoon here. So this means there's a decrease in the number of these tight junction complexes. So the way that the cells are glued together is not as effective and they're becoming a little bit leaky. And what we're looking at is gadolinium contrast agent leaking out of the blood vessels. So the legend here, yellow, means that there's quite a high rate, quite a lot of gadolinium leaking out. Red is not so much. And you can see there's quite a lot of red here and a number of these yellow regions. Below, we see an MRI image for an 80-year-old female healthy individual, and you can see there's a lot less leakage. Now, I should say that, as we'll mention in a minute, that the blood-brain barrier dysfunction at various levels is associated with aging, and that, as I said before, one shouldn't view it as black and white. One should view it as what an individual has the capacity to tolerate, although at some point it may get above a threshold where it can result in symptomatic changes. So this is fairly typical for an older individual. These types of focal leaks are also observed, but at much lower levels in younger individuals as well. Okay, so now we're going to look at a little more extreme defects. So now imagine that we have complete loss of tight junctions at some location, or we have a structural defect resulting in loss of endothelial cells that allows leakage of blood components into the brain, including cells. So we have two MRI images here. In this case, that is from the same individual. The details of the individual were not given in this study, but the mean age was 68 years old, so older individual in this typical example. And in this particular study, they were doing follow-up MRIs to look at changes over time. This is part of the Rotterdam study that was done maybe 15 years ago that's quite well known. So again, this is nominally healthy individual, no evidence of cognitive dysfunction. And in this type of MRI imaging, what we're looking for is dark spots or dead spots that are related to paracellular components. So things like hemoglobin that is, and platelets that are taken up by immune cells. So that's what we're looking at here. And these are known as microbleeds. So these are relatively small, typically a few microns or so in size. And you can see there's a number of these. There's two over here and two over here that are highlighted by the arrows. So the important thing here is in the follow-up scan 3 years later, the same microbleeds exist, but there's also new microbleeds that appear. So
let me ask another question. When you see an MRI like that, and you know the patient or the person involved has not had a stroke, is it conceivable at this stage, when you see these early microbleeds, you could inject some drug that would somehow close the microbleeds or control them and rejuvenate original function?
Yeah, that's a good question, and that's where we're heading. So,
okay, sorry.
No, you're perfectly setting up a lot of this, a lot of what's to come. So that's basically the idea of blood-brain barrier rejuvenation and repair is basically not been a mainstream concept. So everyone's been in the field has been so focused on if you have a stroke or neurodegenerative disease or multiple sclerosis of treating the disease. And there's been very, very little attention that's focused explicitly on the blood-brain barrier. And that's basically what we're focusing on. So it's not going to solve everything, but it's likely to be a very important component of treatment strategies in the future to be able to do this.
Good. Thank you.
And obviously in the context of aging, if one can repair or rejuvenate these types of focal leaks or microbleeds, it potentially could extend the period of time before one reaches a threshold that might lead to symptomatic effects for sure. So all very good questions. I should also point out, as usual, I'm an engineer. I'm not a medical doctor. Don't rely on anything I say, and I try not to say anything that might be associated with diagnosis.
Well, let me just say you could certainly have fooled me because you sound very medical today, and I appreciate that, Pete. Thank you.
Okay, so I'm going to spend a few slides now interspersing where we're going, which Ron has just predicted where our research is going and where the talk is going, along with I'm going to show just a few examples from our research to illustrate what we're doing. So as Ron said, we are a tissue engineering lab. So tissue engineering is a sub-branch, if you like, of biomedical engineering that involves a lot of material science and engineering. So basically what we're doing is building brain-specific blood vessels in the lab. So these days we can be quite sophisticated about doing this. So we typically use stem cell technology to take stem cells, differentiate them into the different cell types into the brain, and then we seed them into a device that basically consists of an extracellular matrix. So you can think of this, if you're a physical scientist, visualize jello as the matrix. That's the main structural component of most tissues. And if you imagine a chunk of jello with a cylindrical channel in it that's very, very small, we line the cylindrical channel with the endothelial cells to form something that's like a vessel. This is in a microfluidic device that we can then perfuse with medium or blood or whatever we're interested in. So this is a microscope image under the microscope of one of our vessels, our microvessels. We can perfuse it, as I already mentioned, the surrounding matrix looks like matrix in tissues. And if we look at the cell-cell junctions, this is a little bit small here, but hopefully you can recognize the features that I showed in the electron microscope images of rat brain capillaries previously. You can see this overlap region here where the tight junctions are formed.
Mm-hmm.
So this, let me see, this isn't, this should be a video, but it's not playing here. I may need to go back. I'm just going to see if I can go back to, yeah, okay, I'm not, oh yeah, here we go. So this is another example of, or this is an example of one of our microvessels that we create, brain-specific microvessels. So the endothelial cells are derived, differentiated from stem cells to be just like brain microvascular endothelial cells. And the cells are genetically engineered so that we can visualize the tight junction proteins that form the junctions between the cells. So the video is going pretty quickly. You can maybe see the cells moving around. So we can put this on the microscope and we can actually visualize the tight junctions. And more specifically, we can visualize the tight junctions as we're exposing the microvessel to different types of stresses or trying to repair defects in the microvessel. Okay, let me go back to the laser pointer here. Yeah, I'm going to skip this example, I think, and move on now to, so we've talked primarily about the machinery of the blood-brain barrier, what it's supposed to do and how it's supposed to work. And we talked about one example of disruption of tight junctions forming focal leaks, so leakage of gadolinium contrast agent or microbleeds. We've looked at MRI images. So as it says here, blood-brain barrier dysfunction is a hallmark of many diseases of the brain. So almost any disease you can think of. So blood-brain barrier dysfunction is associated with all of the neurodegenerative diseases, with vascular dementia, with all of the cerebrovascular diseases, stroke, vascular dementia, I mentioned, cerebral amyloid angiopathy, small vessel disease, brain infections, inflammatory diseases, brain cancer, traumatic brain injury, and even healthy aging. I've showed you some of the MRI images that show there is some level of dysfunction, although non-symptomatic, associated with healthy aging. So this is sort of the big picture to bear in mind. So now we're sort of setting up for the direction that our research is going into. Again, although I'm not going to show you too much in the way of detail. So two things to bear in mind here that's very important with dysfunction. So during life, our blood-brain barrier is subjected to a wide range of stresses. So just to give a few examples, so hypertension, obviously increased pressure is a stress that acts on the blood-brain barrier, if you can think of it as a mechanical stress. Hyperlipidemia, diet, chronic inflammation, aging, and many, many others are conditions that exert stresses onto the blood-brain barrier. So this is sort of a cartoon of how I think about this. So the x-axis would be an individual's life. So the y-axis is stress. So you might imagine you're in college, you play football, you have a concussion. That's an incidence of stress on the blood-brain barrier. Maybe you have an infection. That's going to exert a stress on the blood-brain barrier. Maybe you spend your younger years eating McDonald's a lot, so you have a poor diet for some period of time. That's going to exert a stress on the blood-brain barrier. Maybe you get busy at work in middle life and you have low physical activity. Psychosocial stresses are known to exert stresses on the blood-brain barrier, and hypertension, as I've already mentioned. So this might be a typical sort of life experience local to the blood-brain barrier. So now if we consider blood-brain barrier health as a function of time, so the idea is that we're above some threshold where we can tolerate any stresses or any dysfunction to the blood-brain barrier, and it is transient and can heal itself to some degree. A concussion, for example, may, put us below this threshold. There may be some symptomatic indications as a result of that, but most of the time we're above some nominal threshold that may result in some symptomatic response. Obviously, over time, there is, due to aging that I'll talk more about in a minute, we may get closer to this threshold or dip down below this threshold. So this is the first idea. During life, we're subjecting the blood-brain barrier to stresses all the time. They may be transient or permanent. They may result in some level of dysfunction. They may not. The other thing we need to bear in mind, as I've told you, there are many systems associated with the blood-brain barrier to regulate what gets into and out of the brain. So all these stresses that I've mentioned here are basically associated with disease risk factors. So they can lead to many different stresses, and there are many different potential mechanisms of dysfunction. So all I'm saying here is there's no one magic bullet. You can't say if someone has blood-brain barrier dysfunction, there's going to be one solution or one strategy to repair the blood-brain barrier or rejuvenate the blood-brain barrier. There are many different mechanisms of dysfunction that can arise from different stresses. So this makes it a very complicated problem. And this is pretty much where we are at the moment. So I should emphasize again, this is the current state of understanding. So we're just at the very early stages of trying to formulate these questions and figure out responses. So again, this is just sort of the basic same idea. So if we want to develop treatment strategies to rejuvenate the blood-brain barrier or repair the blood-brain barrier, we need to know what the mechanisms are that are induced by the different stresses acting on the blood-brain barrier, because the treatment strategy is going to be specific to a particular mechanism of dysfunction. Okay, so if we look at Alzheimer's disease and stroke, so of all the diseases of the brain, these are the two that by far and away affect the most individuals and then the most expensive from a healthcare perspective. So as you probably know, for Alzheimer's disease, there's many, many risk factors. There's the so-called non-modifiable risk factors, age, family history, genetics. There are a whole range of modifiable risk factors, hypertension, low physical activity, we've also mentioned, psychosocial stress, hearing loss, alcohol use, as well as other medical conditions like heart and vascular disease. And the obvious connection there is the blood in circulation. So anytime you have peripheral disease that can influence blood in circulation, you can influence potentially the brain. Stroke also has a wide range of risk factors. Many of them overlap. Age is a risk factor for AD and stroke, and aging is associated with, as it says here, accumulation of cell damage, inflammation. You may have heard the term inflammaging or systemic chronic inflammation associated with aging, epigenetic changes, and other things. One of the interesting factors that we're interested in is it's been shown in humans that the composition of your blood or one's blood changes with aging. And there's a concept called the plasma protein clock. So if you were to take a blood sample from an unknown individual, by looking at the composition of the plasma, you should be able to identify that person's age relatively accurately. So this is like, you may have heard of like, you can use telomere length as a way of estimating age. This is just another one of those things that have been studied over the last decade or so. From our perspective, one of the really important things is that it's been shown that the changes in plasma protein composition associated with aging, this is in mice now, but those changes can induce changes genetically and functionally in the brain microvascular endothelial cells, these cells that line blood vessels in the brain. So again, I'll say that again. So changes in plasma protein composition associated with aging, those changes in protein composition can drive genetic changes in the endothelial cells that form the blood vessels. So, so the point I want to make here, again, just reinforce that many diseases of the brain have multiple risk factors. Many risk factors are shared across diseases, and many of these risk factors either directly or indirectly related to stresses that act on the blood-brain barrier. So again, just to highlight, this is a very, very complicated problem. Okay, I'm just going to show a couple of slides of research. I think I've just got 3 or 4 slides to go. So hopefully I'm not boring everyone. So I just want to show a couple of types of experiments we're doing to try to unravel some of these questions. So if we think about Alzheimer's disease, so Alzheimer's disease is associated with generation of a protein called amyloid beta. Although if you've read the New York Times this morning, you'll know that there's some controversy about the significance of amyloid beta because of the failure of strategies to target amyloid beta in treating the disease. But nonetheless, the basic idea, neurons and astrocytes in the brain generate amyloid beta. Amyloid beta activates supporting cells in the brain that generate molecules that cause inflammation, that can act on the blood-brain barrier. At the same time, in an individual may have hypertension or hyperlipidemia. So if one wants to understand how amyloid beta affects the blood-brain barrier, it's almost impossible to do it in an animal model because of all these complicating factors. So an advantage of the types of models that we use in the lab is we can very, very reductively just look at the response to one type of stress. So the particular stress that we're looking at here is amyloid beta. So this video that I'll run in a second is one of our microvessels. So the microvessel is the small region in the center here that I'm pointing out. Flow is from left to right. The matrix is surrounding here. The lighter regions are the polymer plastic housing for the device. And here we've introduced a side port where we can expose to amyloid beta. So the amyloid beta is fluorescently labeled here. So we're exposing our microvessel to amyloid beta, and we can then observe what happens to the blood-brain barrier, so to the microvessel here. So to do that, what we do is we perfuse the microvessel. Instead of with gadolinium, as I showed you earlier, we perfuse it with a fluorescently labeled dye. So this video shows here the sort of magenta color. We're perfusing the microvessel in our device. This is on day 2 following setting everything up. And we're not exposing to amyloid beta here. And you can see the dye is contained within the microvessel. The tight junctions are doing their job. Now, if we repeat the experiment on day 2 after exposure to amyloid beta, so just like I showed up here, we're exposing the microvessel to amyloid beta. So if we repeat the experiment, and you can maybe make out there are focal leaks along the length here. It's a little bit difficult to see because we're seeing an overlay of the microscope image and the fluorescence image, but it should be evident in this photograph over here. So amyloid beta induces focal leaks in the microvessel that, again, we spend a lot of time now studying this. They're transient. We're looking at concentration dependence and so on. But this is just one example about how we can deconvolute all these contributing factors so we can just explore how amyloid beta alone influences blood-brain barrier function or dysfunction in this particular case. I'm going to skip the next one, and I'm just going to go to the final one. So two slides left. So I just want to show you another example here of a type of experiment we're doing. Same basic idea, very reductive, of trying to figure out how very specific stresses influence blood-brain barrier function. And this one is related to stroke. So a hallmark of stroke is stenosis, so narrowing of a vessel. There can be many reasons for this. And basically, we've developed a way to mimic this. So in the video here, we have our vessel, which is vertical in this case. The cartoon here illustrates what we're doing. So what we're doing is we have a probe that's going to come in from the left, and it pushes the matrix, which constricts the vessel. So here you can see the probe coming in. Here's our microvessel. Flow is from bottom to top. And you can see the probe that's inducing a stenosis here. It's inducing a narrowing of the vessel. So again, this is very reductive. We can do this in the absence of hypertension, in the absence of plaque buildup, in the absence of hyperlipidemia. So here we're asking the question of how stenosis alone influences blood-brain barrier dysfunction. So you can see from the images down here, with no stenosis, the yellow dye here called lucifer yellow is contained within the vessel. There's no leakage. But when we induce 70% stenosis, you can see there's a number of these focal leaks. So there's a loss of tight junctions transiently along the vessel here. And we can do this very carefully. We can look at how where individual focal leaks occur and how that changes when we increase the stenosis to 90% stenosis or 100% stenosis. Again, I'm not going to go into details, but all to show the general approach. So here we can look at the effects of stenosis alone. We can then build in inflammation. We can add inflammation and see how the combination influence blood-brain barrier dysfunction. We can introduce other factors as well, and so gradually build up the complexity. The important thing in the bottom line, though, is now we have well-defined models that we can test strategies for blood-brain barrier rejuvenation and repair. And these haven't existed before in this kind of reductive way. Okay, so let me just conclude. I think I've gone on long enough. So the idea of blood-brain barrier rejuvenation and repair, as it says here, not a mainstream concept, not something that very many researchers at all, in fact, very, very few researchers are thinking about. Part of the reason for this is it's so complicated. As I mentioned, there's many mechanisms of dysfunction that can be caused by different stresses. And so you need to be able to have some sort of reductive approach to be able to do this. So to develop strategies for repair, you need to identify the mechanisms. This just gives you an idea, I apologize, it's very small, of the kind of things that we and a few others are thinking about. So potentially there are drug therapies for rejuvenation and repair, potential non-pharmacological interventions. So hypothermia therapy is something that's used experimentally in stroke, for example. Diet also has huge potential. And then cell therapy and gene therapy also have potential in rejuvenation and repair. So basically, in conclusion, I think the take-home message for you guys and ladies and for me is to look after your blood-brain barrier.
Thank you, Pete.
So yeah, I hope that's been informative and that you've learned something about the blood-brain barrier.
Pete, thank you very, very much. You know, to say that that was informative is a tremendous understatement. It was truly brilliant. I learned more than I ever could imagine in your presentation today, and I appreciate that. I just have a couple of questions to begin. Maybe you could stop the screen share and we'll get everybody back on the line. You know, as an engineer, many of the people in your audience have had a design experience. But when you look at the design of the blood-brain barrier, you have to marvel at what nature has done. I mean, can you imagine an engineer trying to design something like the blood-brain barrier, and yet nature has done it for us? It's truly remarkable.
Right. So the one thing I would say here is there's a neuroscientist at Hopkins called David Linden. Some of you may have read one of his popular science books. So he likens this from a design point of view. I think you guys who've worked in aerospace and related industries will appreciate this. So, you know, we've evolved this unbelievably complex system that we have to power the brain, which is hugely complicated, but at the same time, we have to protect it. And if you had to design it from scratch, you probably wouldn't design it the way it's designed, right? We have 600 kilometers potentially for failure, right? Is not the way you'd want to do it. So Dave Linden likens this to, or the way that we've evolved to, if you were flying an aeroplane and you were trying to, while you were flying it, you're trying to upgrade it. So you start off, say, with a very simple propeller plane, and then you develop jet engine technology. So he likens it that you're trying to install jet engines while you're still flying the plane with propellers and introduce electronic circuits instead of, you know, pneumatic circuits to control everything, for example?
Well, there are a number of hands up, and I'm going to turn to them, but I just want to say, what you mentioned in your closing slide that your work may not be mainstream in terms of what people are doing, or I think that was the language you used. But I want to say, I think you're on a path that, from the perspective of an aging population, could actually be absolutely revolutionary. It, if in fact many of the dysfunctions associated with memory loss, for example, are associated with leakage of the kind you've described, and you can find a way to control that leakage, that will change life for many, many people throughout the planet. And I just want to say that I think you're doing some really remarkable stuff. But let me turn to the questions. Tom's up first.
Yeah, this was a remarkable presentation. I really sort of learned a lot. One question I have here is, you showed on your chart, the regeneration and repair, sort of this very highly nonlinear relationship between sort of inputs and responses in this system. There's a lot of redundancies, I think, that get involved in there, et cetera. But has there been any efforts yet, or beginning efforts essentially now, to sort of try to unravel these nonlinearities using AI and AI techniques? What you actually showed in your picture was sort of a typical kind of AI system, learning system. So has there been any efforts essentially to use these more modern techniques to look at this from sort of, let's say, an analytical rather than an experimental approach?
That's a great question. To my knowledge, nothing has been done. And the main reason for that, I think, is just the lack of data. So, as I outlined, it's a hugely complicated system. There's multiple stresses with multiple potential outcomes. And one of the challenges for scientists in biomedical research, as you guys probably know, is there's actually relatively little data to compare to.
Yeah, okay.
So if you're working with animal models, you have a huge amount of data available to you, but the ground truth is what happens in humans. And we're very limited in what we can do for obvious reasons. So the amount of data that we have to connect to, whether you're working with animal models or, like us, you're working with in vitro models, there's very little to compare to.
Okay. All right, thanks.
Let's go to Jenny. Jenny, you're muted.
Say, if we say that our thoughts or any other cognitive functions is kind of result of a dynamics of neurons. If we accept there are 86 billion neurons in an average human brain, and then we have all this electrochemical signals zooming around in the brain, which really create all these cognitive functions. So with this barrier, as you illustrate, very, very intrigued mechanisms, is there any study about how the neuron dynamics really related to the barriers functioning or dysfunctioning, as you just illustrated with those research, and how that really correlates to basically what are the findings if there's such a study, and how they really affect our aging, when we have a decline in the cognitive functions. Basically, I'm trying to correlate this neuron signals transmitting, in the brain and versus this physical, biological or barrier.
Yeah, I wish I had a better answer for you. Unfortunately, I don't. I'm also not a neuroscientist, so we don't do any work with neurons. But I think the sort of picture I have, as I alluded to, is that the neurons need to work in a very, very tightly controlled, pristine environment. And if the local environment is perturbed or becomes more variable and sloppy, then it's more difficult for those electrochemical signals to propagate cleanly and along or across synapses and so on. And I'm not sure that much is really known in detail about how the electrochemistry of the environment, right, contributes, or how much fluctuations one can tolerate to limit signal degradation. So in fact, you guys, I think several of you said that you were in sort of signals and communications at various points. I think that may be a good analogy, right, in terms of how much degradation of the signal can you tolerate in terms of propagation of action potentials if the environment becomes more messy. So I'm not sure I'm answering your question, but I'm not sure actually that much is known about it.
Thank you.
Eddie?
Yes, I've actually got a lot of questions. I will say I very much appreciate the fact that you're bringing finally a complex system down into something that is handleable experimentally. That I think is wonderful. My question is, within the brain there are microtubules, and if you start loosening the brain barrier or opening it up, then bigger things will get through. And I wondered if you have any understanding of what happens when bigger molecules start getting through and could block up the microtubules, for example. And the other question I have, when you stress the capillary of the blood brain of where the stuff is flowing, it seems there are two ways in which it seems like there can be leakage. One is by actually, because you create a curve, you extend the thing, and so there's more, a gap opens up. Or is it that the elasticity of the thing actually becomes plastic and it never goes back again once you release it?
So the answer to that is both. So I simplified a lot of aspects here, but basically there's possibilities for both to occur. Basically, I'm not going to go in, I don't think it's productive to go into the details, but both can occur. But I also want to emphasize, I talked a fair amount about leakage between cells. I also mentioned, though, that glucose gets into the brain via carrier-mediated transport. So there's specialized proteins that grab glucose from circulation, pump it into the endothelial cells, and then it's pumped out into the brain. So another mechanism of dysfunction is if there's dysregulation of those proteins, or if there aren't enough of them on the cell membrane, or they're somehow not working properly. That's another possible mechanism of dysfunction. So like I said, there's many possible ways that things cannot work properly.
So one of the issues I think for me is if the brain is stressed, or there is greater stress in the human body, blood pressure, that kind of stuff, these arteries get, or these, I'm sorry, I can never remember what these things are. I've got a rotten memory for names. But basically, when you stress the capillary, if it's plastic deformation, then it doesn't go back. So over aging, you basically have a problem. But if the stress is released in other cases, and it's just just simply a kind of deformation that can be relaxed back, then it's not an aging process. And I've just wondered if over time, do you have any evidence that this sort of process occurs, that it continues to just simply build up, and that's what we call aging.
I think in essence, you're correct. There are mechanisms by which that often occur with aging, where especially the larger pulsatile vessels, so the arteries and arterioles in the brain, can become stiffer, and that can reduce their ability to dilate for sure. From what I think is known, there's huge variability between individuals as well. And there's also potential, regional differences in the brain. And then it's a question of, how prevalent it is and loads of other factors as well.
Right.
Some other interesting things is, weightlifters in particular apparently have huge transient increases in blood pressure in the brain in terms of their, but that apparently leads to no symptomatic effects. So these things are reversible, and as I emphasized, whether they're transient or persistent, and how extensive throughout the brain, and a lot of other factors contribute, I think, as whether they might cumulatively result in something that could eventually be symptomatic.
Yeah, my big question mark is what does aging actually mean? So I'll leave it at that.
Yeah, I think that's a big subject of research, that people are still trying to figure out what so-called normal or normative aging is characterized by.
Or what even is aging at all? So anyway, I'll leave it at that.
All right, let's go on to Avi. Thanks, Eddie. Avi?
Yeah, a really great presentation. I've got three quick questions. One, you mentioned diet, and if you have any tips as to what we should eat or not eat with regard to helping the blood-brain barrier, I'd be interested in hearing it. The second is, we talked a lot about repair. Has there been any thought given to something prophylactically that could help reinforce or ensure that these micro leaks don't occur? That, in other words, reinforce the linings, or I'm not sure the terminology of what so that the chemistry would be different. And the third is, there's some cancers that I understand metastasize to the brain. Others don't. How do the cancer cells get through the blood-brain barrier?
Okay, so all really good questions. So the first part, I think, in terms of prophylaxis, I think what's known at the moment is all the things that we're all aware of in terms of healthy lifestyle is going to help. So, exercise, minimizing red meats, balanced diet, all those kind of things are going to help, because the same risk factors, as I mentioned before, risk factors for almost all diseases of the brain, there's many overlapping ones, and we know how to, at least the lifestyle ones, we know how to mitigate. Your last question related to brain cancers is a good one. Breast cancer is one of the cancers that metastasizes commonly to the brain. So tumor cells are shed from the tumor into circulation. They travel throughout the body. Many metastatic cancers occur in the next organ along from the source organ where the primary tumor is. And so those tumor cells, in some cases, are able to stick to the microvessel wall and burrow their way through into the brain. But in many cases, it's likely to be clusters of tumor cells that basically get lodged in a small capillary. That causes a region of disruption, like stenosis in stroke, that gives rise maybe to weakening of tight junctions and makes it easier for the tumor cells to migrate into the brain.
Okay.
Thank you.
Thanks, Pete. Albert?
Peter, you said that the brain consists of a huge network, 600 kilometers, etc., and that it is vulnerable in many places. Am I correct in thinking that given that it is such a large network, it would be more forgiving of failure to happen in one place or the other, and that it's a network, therefore other connections can take the place of what has gone wrong? What do you think of that interpretation?
So again, you guys are asking very, very broad um, questions. They're all very good questions. So let me answer that like this. So during development, when we're younger, as we're generating more neurons and more circuits, the vasculature in the brain is also adapting, creating new vessels and so on, new capillaries to supply those neurons. When you become an adult, there's generally very, very little change in the vasculature in the brain. It's more or less fixed, but there are situations where one can create new vessels. So, for example, high altitude adaptation. So, if you go from sea level to high altitude, because of the decreased oxygen function, you can generate new capillaries in the brain to provide more oxygen to the neurons. There's also the reverse case. So studies in animals know that if you take a mouse in a so-called rich environment, so in a cage with a treadmill and so on, and you place it in a in a poor stimulatory environment, then it will decrease. The number of neurons will decrease, and the vessel of capillaries will decrease as well. So this is known as neurovascular coupling. So in some ways, the number of vessels adapts to the number of neurons. But normally throughout adult life, there's very little change.
One hear about the plasticity of the brain. That stops at a particular age, or what?
Again, now we're getting into areas that I really don't want to say too much about, but brain plasticity is more related, I think, to at the cognitive level, our ability to do different functions in different regions of the brain. So I'm just focused on how to power the brain. So the vasculature that powers the brain and then protects the brain and allows it to work in a normal environment.
Thank you.
Thanks. Let's go on to John. John, you're going to have the final question for today.
Okay. So this is maybe more of a comment in relation of the talk today to a number of other talks we've been having recently focused on AI. And one of the comments that was made in the talk was that the brain we have was as a result of, thousands of generations of evolution through different, stages of development and into humans. And so certain of the things might have been designed better, but to make changes evolutionarily, it's like trying to fix a jet engine while it's running. So we're kind of like stuck with what we've got. It occurs to me we've heard a lot of somewhat scary things about AI, and I'm even more scared now because AI is not limited like that. They just can build more and more computers and connect them however they want, and et cetera. So that's just an observation. I don't.
Yeah, I think that's a good point. I think the issue would be one of feeling that you've got trusted data you can use to feed into your AI learning machine. And that's, I think, where many of the problems remain. Anyway, this has been a wonderful conversation. I want to say a couple of things. First of all, Albert, I am happy to see you today. I really am. I know that having thyroid surgery is not a exactly easy thing to deal with, and you look good. I'm glad to see you. I'm really happy that you're online today.
Thank you.
The lady next to you, at least on my screen, my wife Carolyn has had thyroid surgery, so she knows about that. And Pete, I want to just point out that the second half of your extended academic family has been online during your presentation. Carolyn is here, so you guys may want to say hi to her.
Yeah, so if Carolyn's there, please say hi. I remember fondly coming for many potluck dinners at your house that Carolyn hosted.
Yeah. Go ahead.
Unmute myself.
You're muted now. You are muted, but you can't unmute. There you go.
Just want to say hi. It's nice to see you after so many years.
Yeah, it's unbelievable how long it's been. Yeah.
Yes.
Well, we have to get you back up to Boston. That's the solution here. But we'll come back to that on another occasion. Let me just close by saying that, we began by putting in context that this was another illustration of the emerging and developing interface between engineering and healthcare. And Pete, you've given us an absolutely brilliant conversation and presentation on something that's of concern to all of us. We're all aging. As I said, nobody's growing younger. We're all aging. And I think what you're doing is truly revolutionary. And frankly, I hope I hope as much as I can hope that you're successful, because I know that if you are, you're going to help a lot of us who are online today, and you're going to help a lot of people all over the planet. So, number one, I wish you lots of good luck. I thank you for being with us today. And I just want to bring the session to a close with loud applause to our speaker today. It's been a great presentation, Pete. Thank you.
Yeah, well,
Take care, everyone. We'll be back together in 2 weeks. Pete, did you have a comment? I'm sorry, I may have cut you off.
No, I just wanted to say thanks to everyone for being engaged, and it's always great to get really good questions and food for thought. And just to remind everyone, eat your broccoli.
There you go. Thank you. All right, that's our closing messages. Eat broccoli. Thank you. Take care, folks.
We had it last night.
There you go. Take care, everyone.